Good morning, everyone. Thanks for coming along to our RIEL Seminar Series of May, 1st of May. So I think everyone's been here before, but just a little bit of housekeeping. If there's an emergency, we'll go out to the grass out here, and bathrooms are just under the stairwell. But today we are welcoming Padraig, who is a senior lecturer with Menzies Medical Program here at CDU, and he has a background in biomedical sciences and virology. But today he's going to be talking to us about crocodile carcasses. So that's where the link is to RIEL, and looking at a bit of crocodiles, which I've got my key earrings on to support today. So if we want to give Padraig a round of applause, and I'll pass it over.
Hey, thank you very much. Thank you for the invitation, and hello and welcome to everybody online. So this is a fairly familiar environment for me, teaching in a hybrid space. So I'm going to talk about our research around adding value to the crocodile industry. First came across the crocodile industry when I was at Central Queensland University. And there's a crocodile farm run by a very innovative farmer called John Lieber outside, and we got talking about the industry and its challenges and opportunities. And one of them we thought about is what we can do with the waste carcass after the valuable skins are removed. So that's the focus of today's talk, the research we've done, and how to really look at products that can be developed that will really add value in higher end biomedical applications. The second bit is a bit adventurous and really I guess prospecting as to what a bioindustry might look like in the NT, and how collagen really might kickstart a biomanufacturing, I guess, ethos or ecosystem in the NT. Okay, so first I'd like to acknowledge all First Nation people across the lands with which we live and work, and pay our respects to elders both past and present. Okay, so I've slightly rewritten the title now, as you can see, only after 3 slides. So this is really a title of a grant that I submitted a couple of months ago. So developing a Northern Territory biomanufacturing pipeline to include both crocodile and barramundi. So barramundi industry is very interesting. We have one of the largest barramundi fish farms in Australia, and they have a very interesting waste product, which is the swim bladder, highly collagen dense, very high grade collagen. And the crocodile complements that with very different types of collagen from the range of different parts that can be available from the carcass. So we're unique in the NT in terms of the collagen industry, and we need to articulate that. Okay, so hopefully this will help. So what I'm going to talk about is the work that AgriFutures funded us. But this is my favorite street art, just opposite Woolies in Cavan Street, is about processing the waste. Primarily, we looked at the tail and also the ribs, costal ribs, and really purifying and characterizing the collagen and being confident that we had collagen peptides and identifying them then. And then the applications were to look at really the higher end of biomedical applications in terms of wound healing. Okay. And really trying to understand how this links back to behavior in terms of the resilience of the animal, how it survives certain things in the wild. Wounds obviously are part and parcel of the crocodile's life, and it seems to survive incredibly well, and in part regenerate as well. So that's croc collagen. Then I want to introduce you to something really interesting, is how we use the oil of the croc. So there's a small number of companies. One of them is called Golden 8. They're located in Melbourne, and they've developed croc oil products with very little science behind them. And that's the problem with small, relatively small cottage industries. Very passionate individuals about the product, but don't have the science. So we hooked up with them. And I happened to have a fellowship in Korea at the time, and I worked with people in the cosmetics industry about looking at croc oil, particularly on anti-inflammatory effects in dermatitis. So I think that's really interesting, the high value potential there. And if we get to it, I really want to, I guess, propose what that bioindustry would look like in the NT. All right. So this is where I was last year, the guy in the shorts. So I just arrived to start at the new CDU Menzies Medical Faculty, and that was a strong drive for me to come here, the chance to start, in a new development, a really brave approach to teaching medicine to equip students properly to serve the community, to be culturally responsive, to be aware of the health challenges in this region. And so we've got 43 this year. They've just started, so it's really full on. This picture now is doubled in size in terms of our staffing. But what's fantastic is that we're such a culturally diverse group of people in the medical school, and it's a fantastic place to work. Okay, so usually in a pitch you ask at the end. So I'm going to ask two things at the start. Okay. So the academic opportunity was about working in a medical school, and the scientific opportunity was to follow on what was funded research in the crocodile industry. And so in between all that, I'm a teaching academic, and right now it's incredibly hectic because I'm doing what's called the HEA Teaching Fellowship, and alongside I'm also doing the CSRI on Prime Research Translation Program, and that's also hectic. So both of them require, workshops each week. So in between there's things that we can do together. Okay, one of them revolves around teaching. Okay, and that's how I really would like to talk to people within RIEL and environmental sciences about developing a resource, either a lecture, a workshop, or it could be an online resource around One Health. Okay, particularly for our medical students. And I think it's something we can do really well because we're so co-located. It's not something that's often taught in medical schools. And particularly when you look at that triad of One Health between animal health, human health, and the environment, it's always the environment that's neglected in the triad. It's always a difficult one because it's a confounding dynamic environment and how we associate with disease. So particularly ideas around climate change and antibiotic resistance, I think is really pertinent to teaching medical students. So that's my ask. If you're interested, contact me. I'll send an email out again and we'll meet and we'll think about how we can formulate this, for next year or something like that. The second one is about CSR on Prime, which is an amazing experience. It's pretty intense. We have a team called NT Biocollagen. There's myself and a pharmacist from CDU called Nazim Nazar. And we have two members from my previous lab in Perth. And we've just come back from Perth from a week-long intensive. So we have 10 weeks to really refine our research in terms of translation and economic viability. And it's asking tough questions. And we do that by carrying out 100 conversations. Okay, we have to speak to 100 people broadly around the industry that we're trying to solve a problem for and those stakeholders. So you are important stakeholders in the crocodile industry, in any change that happens in the NT, whether it's agriculture or a new biomanufacturing. So that's my ask, is that I might need to talk to you for 15 minutes. It's a very structured conversation. It's not about research. It's about what are your challenges and what will be the impact of what we propose in terms of your environment, okay? If we do 100 conversations, CSIRO will give us $5,000, okay, at the end of it. All right. So the next few slides really just highlight what I was talking about in terms of how we would formulate that. All right. So I'll quickly go on to what we want to talk about today. All right. So this is an online report that you can look at that was published by AgriFuture, and so we looked primarily at farmed crocodile, so not wild crocodile. And I have to admit at this stage the crocs were from Queensland. And I did bring the majority of the isolation and characterization was carried out in Perth. So very keen to do some similar work here in Darwin and having a lot of interaction with PRI farming at the moment. All right, so let's talk about collagen, a little bit of biochemistry. So collagen is a triple helix type structure. It's formed of these 3 polypeptide chains, and that's my best 3D printing representative of a collagen triple helix. And you'll see there's the color-coded, this tripeptide, and there's a constant repeat of proline. And that's really important, okay? So when you think collagen, think about mechanical strength, okay? And so we have proline plus 2 other amino acids. They're primarily, glycine plus proline, and then we have a proline that's hydroxylated. Okay, so the hydroxylation is important because it means that the protein is highly modified post-translation. And that's going to link back to something I talk about when we look at collagen, how to make collagen outside of animal systems. So the basic unit is called tropocollagen, which is the triple helix. And then that's arranged into these fibrils up here on top right in a kind of head to tail arrangement. And they're packed really densely by cross-linking, by hydrogen bonding, or covalent linkages between the lysines. So the high concentration of those amino acids, such as protein and lysine, are there for a reason. They're there to ensure strong linkage between the fibers, because once you have the fiber, or the fibrils, they're then linked to form the fibers. Okay, and so that the density and the pattern of that linkage, it represents where that collagen is in our body. Okay, so if we think about our tendons, our muscles, we'll have tightly packed collagen fibers, adding to the mechanical strength and the load that's needed in those tissues. If we think about collagen in our face, our skin, they're a lot more loosely packed. Okay, and what happens to collagen as we age is very obvious, that we start to lose some of those connections and our skin starts to sag. And that's obviously one of the prime, economic drivers of using collagen in the cosmetics industry as well. Okay. So besides structure, type is important, and type represents different parts of our body. Collagen type 1 is the main type, which we find in the majority of tissues. But we have interesting types, particularly around articulating joints, in our knees, our elbows, etc. It's called type 2. And that's one of the reasons we went after the croc, because it's a very difficult type of collagen to harvest from the major primary sources, which are the cow and the pig. And so the croc has interesting articulating joints, to strong weight, load bearing. And I think evolutionary, there's a thicker layer of hyaline cartilage in the croc as well, which would represent a greater source for type 2. And then there's type 3 around the vasculature and muscle as well. And so, as I said, how it presents itself in the body, it has to do with how it's densely packed in terms of its fibrils and the function it has in that location as well. Okay. All right, so why collagen? So I think the next time you think of the crocodile tail, that amazing mechanical force that comes from the crocodile, think of collagen. Okay, so think of how densely packed that collagen needs to be to withstand the forces that are transmitted through tendons and ligaments during swimming or jumping, etc. And we don't see that in any of the other production animals, obviously, where collagen is sourced from. So there's a very unique source of collagen that's tightly linked to function. Okay, so if you think of more sedentary animals, where we get collagen in the cow and the pig and the sheep, they're not doing this. Okay, they don't have attributes in their body where collagen needs to be as tightly packed together. So there's something unique about the location and the structure, and as I mentioned, also interesting parts around articulating joints as well. Okay, and in the industry, it is incredibly efficient in terms of the other parts of the body, but primarily, obviously, the skin and the underbelly skin is the prime bit, and a lot of meat comes from the tail. But there is opportunity to harvest quite a lot of material from the tail as well, which would have a lot of collagen. Okay, all right. So again, you can't go into somewhere like Chemist Warehouse or your local supermarket and you'll see a lot of collagen products, and they sell for quite an exorbitant price. So the latest figure I could get was, it's an incredible market of over 10 billion in terms of collagen. And the collagen market is stratified between lower value and up to higher value. Collagen is incorporated in lots of things, in foodstuffs, in cosmetics, in nutraceuticals, as we can see on these shelves. And then the higher value, it's very important in biomedical devices, in biomaterials, and in the new medical approach in terms of regenerating tissue, making scaffolds from collagen as well. So when you're thinking about positioning your research to be economically viable, you're thinking about what stage your product could get into. So we're particularly interested in wound healing. And that's what I'll show you is the application of the collagen peptides that we discovered and used around wound healing, which would take a lot longer to get to market, but the value is much higher. And that might suit somewhere like the NT, where would have a potentially low volume, but high value product in that stream. Okay. So these are just some examples. You'll see them in collagen gummies, various sweets, etc. Okay. All right. So when you think about peptides, this is really that large tropocollagen having been chopped up by pepsin or other enzymes that'll break it up into short peptide sequences, and then they become functional because they can become absorbed better in the body. So in terms of the smaller peptides, are more bioavailable and more functional as well. So currently, the largest source is the cow and the sheep, but there's a very interesting source from marine, which is from marine waste, and that tends to get a higher value. Typically, that is perception of a cleaner source, but often it's waste. Okay, it's skeletons, it's heads of fish, etc. There's an even higher value one from jellyfish, and that's called collagen 0, and that's a very interesting collagen as well. And there's a really innovative company in Wales that started some time ago, and they're really working, in a very special type of collagen that comes from the jellyfish. I'll show you a picture in a bit that highlights all of the publications to show you the range of species where collagens come from. Okay, so let's start with the process that we developed. On the right is Rena Wong, who was the postdoc that carried out all of the work and did an amazing work. Rena's also an incredible communicator, author, songwriter, performer. And we came here 2 years ago to the Crocodile conference. There was a specialist meeting, and Rena prepared a song for the conference. So not an area that I can compete in. All right, to harvest collagen then, we obviously have to trim the parts. So we said we worked mainly with rib. So we would remove the flesh. We would then demineralize, soak those bones in EDTA over a couple of days. And then the typical extraction involves acid. So acetic acid will liberate collagen from the cartilage. And then we add in pepsin, which will start to break down the collagen into smaller parts and increase the release of collagen from the material. So you reach large volumes quite quickly, and then you have to obviously precipitate out your protein using salts to try and harvest that collagen. So it's quite labor intensive, and then to purify involves freeze drying, etc. So that's the lab scale thing. And what we want to do in our next grant is try and get to a pilot scale in terms of increasing scale. Okay, so this is Rena's video link to show you various things that happen. Okay, and there is meant to be, anyway, you do see her stirring it as well. And this actually is one of the largest preps of collagen that we received, and you can hear us crunching that. So imagine kind of crunching sound. So at the end of the process, after the precipitation and drying, we'll get a freeze-dried mass of collagen. That this is what we can work on in terms of solubilizing for functionality. But in the industry, that would be ground up into a very fine powder, and that's what you would buy in retail. Okay. So from these hard tissues, generally we get a yield of about 10 to 15%, which is pretty average. So if you can source collagen from softer tissues, you'll get a much higher value. So hence the reason why I want to look to the swim bladder and get a much higher yield and try and balance really unique collagen sources, which will have a lower yield in our process, to higher yield, which maybe have a lower yield, a lower value application. Okay, so in identifying collagen, then we first look at structure. Okay, so we use FTIR to vibrate the molecule and give us a typical fingerprint in terms of its vibration. So using this machine, which is fairly standard in a biochemistry lab, we can then get a typical FTIR spectrum, which will give you a fingerprint for collagen. So that can be compared then to other sources of collagen. So we would compare that fingerprint to bovine, to rat, to pork, to make sure that we see the typical peaks. And one of them is the amide peak, and that represents the triple helix of collagen. Okay, so we're pretty happy with that. And it meant that we had kind of pure collagen prep as well. The other thing that we do is called differential scanning calorimetry to look at the melting point of collagen. So that's the temperature triple helix falls apart. And that will be defined often by species. Species will have different TMs as well. This just represents the fairly expensive and lengthy experiments we had to go to get this graph. Okay, to get a number against crocodile collagen. But in fact, that's the first of that. That's the first number described for saltwater crocodile, at least. So we have a Tmax of 74 compared to bovine, and that really represents the molecule itself in terms of amino acid differences. It's not really representative of, say, habitat or environment. It just tells us that there is something different in terms of the sequence of amino acids, which is interesting because that would suggest a potential difference in hydrolysates or peptides that we make from that. So the other confirmatory tests are to take that protein and run it on a gel, and it's something that you would typically see is that kind of pattern, which again represents the different forms of collagen, the higher structure in terms of triple helix, and then when it's pepsin digested, we get smaller bands, and then confirming that with something called Western blotting with specific antibodies to collagen. The table on the right then represents a much more sophisticated way to confirm identity. That's by mass spec, by taking those peptides and identifying them then through mass spectrometry. And that we outsourced, but it gave us a hit immediately around saltwater crocodile. So we really have to go through these steps to ensure that what we have is truly collagen. The interesting thing is, so these are examples of a number of 50 type of peptides that we would find in that gross digest, and they're mainly from collagen type 1. We did see some examples of collagen type 4 and type 5 as well. Okay, so a mix of collagens from that preparation. Okay, we can do other things that are characteristic, such as a UV plot, but these are all the standard tests that we would have to do to publish this material to show that it's collagen. The table on the right is interesting. It relates to what I was talking about in earlier on, and again, it confirms things like high levels of proline and lysine, which are typical of that tripeptide repeat that we saw in the triple helix. And the characteristic proline that you see predominantly in collagen is the hydroxyproline, the hydroxylated form of proline. So all of that contributed to the characterization. So from that, we went on to look at function. And we looked initially at what this material looked like under transmission electron microscopy. And this is where collagen is just spread onto a surface. And you can see that at higher power, we can see this dense intercalated network. And that's what we wanted to see when we coat substrates. Coat surfaces, excuse me. So we want to look at cells attaching to collagen, particularly in an assay called an in vitro wound healing assay. We would take typically, you guys don't do cell culture. So this is a tissue culture flask. It's simply a treated plastic flask that we would grow cells. And we grow a cell line that's called a HACAT. It represents our skin cells, keratinocytes. So we grow them, not necessarily, we grow them in a dish, but we create what we call an artificial wound by simply scratching through the monolayer. So once cells have formed a carpet of cells, we can scratch through with a pipette. And then we can add our test substances or at least have collagen on the surface and see how quickly that wound is closed. And we can do that by imaging, real-time imaging, and then quantitate the speed of closure. And that's a very typical assay for wound healing in research and industry. Okay. So this is, again, represents what happens in real time. You can see on the left, we have the plastic that's coated with collagen from the croc, and on the right is the control. And over time, over 24 hours, the live microscopy will compute the number of, the speed at which it fills the gap, essentially, and the increased number of cells and so when we do this over many times, we see the collagen coating being superior to controls. Okay. All right, so that's typically how you then quantitate it. So the imaging is live over 24 hours, but the imaging system will take grabs from that and then quantitate. Are you serious? Right. Okay, we better get a move on then. Okay. All right. So statistically, they was very significant. We did another assay with impedance, which tells us that again can be more quantitative about the concentration of collagen. So I'll just move quickly through to the oil, okay, because I think this is very interesting. So this is looking at atopic dermatitis, okay, which is an acute inflammatory response, and we teamed up with Golden8 and we did this work in Korea, which involved these keratinocyte cell lines firstly being exposed to a stimulant, an inflammatory environment called a cytokine, and then we add the oil to see what other molecules can be reduced in that inflammatory environment. Okay. So the first piece of data is really encouraging because the oil has absolutely no toxicity to keratinocytes. You can put as much oil on these cells and there's actually no harm. This graph then shows you the production of an inflammatory cytokine called RANTES. And as you go from control, you can see high levels. As we move from left to right, you can see that we can completely almost reduce RANTES production by using a 10% solution of crocodile oil. And we confirmed that with another one called TARC, which is another inflammatory cytokine, and that can also be reduced at 10%. So between 10 and 1% of crocodile oil can, in this standard inflammatory assay, reduce these cytokines. Okay, and this is really encouraging, but the really depressing part is that everybody else is doing this, okay, and not in Australia. And so in the last couple of years, this interest in crocodile oil has really heightened. You can't see that very well, but there's a paper from mainly from Thailand, which is a strong crocodile industry, and also from China. And at the bottom is the fatty acid profile of these species, and that's very different. And so I think there's a real scope for us. I'm sorry you can't see them, but there's a real difference in oleic acid, palmitic acids, and linoleic acids between species, and yet we don't know what's in the salt water. Okay. So I think we have something quite special there, but we need to characterize it. We need to put science behind it so we can protect its IP. And what we would like funding to do is to take the waste and render the oil, and then identify by mass spec those fatty acids, and then go through a series of tests. And we can then formulate the oils, which will add value to our IP as well in terms of nanoemulsions. And we have this expertise here in terms of pharmacy from nanoemulsions. So it's something that's achievable in the NT. We can definitely identify by outsourcing the mass spec, but we can make these products here and really add value to this, but protect it so that we can hopefully seek investment for spin out. The other part that I'm really interested in is the stomach. If you think about how this crop can withstand, I guess a huge microbial diversity that's pathogenic and non-pathogenic, and yet there must be something very special about what makes up the mucous membrane that separates the epithelial cells from that hostile environment, and particularly these what we call bottle brush structure. That's it. They're mucins, and yet we know nothing about the mucins that are in the crocodile, and they're probably quite unique in terms of the number of their architecture and the number of carbohydrate chains, etc., that are associated. Because the stomach's a very interesting part of the crop, because we get increased blood supply to the stomach during feeding. Okay, all right, but this advocacy for adding increased biomedical research is nothing new. This comes from our ABC article where Graham Webb again was advocating for this in terms of what we have in the NT. So I think we need a renewed push in terms of protecting sensibly and smartly IP so that we can seek investment. All right, so very quickly, Rena was incredibly good. We actually had some fun about making cosmetics with crocodile collagen, which people are very interested in. So the next stage is about scaling up, which is part of the AEA grant, and then linking up with Pompidou Barramundi to look at another source in terms of type 1. Okay, and again, using those high-end analysis mass spec to identify peptides and link them to function. Okay, so this is the on-prime team. There's 3 of us there, Naz, myself, and Rena in the middle, and our team is NT Biocollagen. So the last, I hope I have 5 minutes, 10 minutes.
2 minutes.
All right, so I'll go a bit slowly now. So the word bioindustry kind of pushed me out of my comfort zone. I really had to think about how this is going to work in the NT, and how we can do this in a stepwise manner and really bring the industry on board. So what we can do is encourage industry to start to look at other byproducts, and not from 2 perspectives. One is to add value, an economic value to their return, but also to improve their industry in terms of a circular economy. Okay, so we're using more of the croc, and that's obviously going to be advantageous to them. It's advantageous to the environment. It's a better perception of the industry that's being efficiently run. So if we add in oil and crocodile and collagen to that, there's a much greater use of the carcass that's left, and that adds, and we can enter markets hopefully in the future that would certainly add economic value. And something like the oil, it's what's called low value valorization. So there's low amounts of it made, but it's of incredibly high value, particularly if it's applied to biomedical applications. And so at the moment, the fat is thrown away. It has to be, it costs to get rid of that fat. So if we bring this into the equation, we're paying for that essentially, even if it's a relatively low value to start with. Okay, and then if we think about bringing in multi-species into that equation, particularly around high yield sources of collagen from the swim bladder, we can begin to think about something like a shared bioprocessing hub, something that both industries, at Croc and the barramundi, and perhaps bovine industry in the future. But what this will represent is a focus of infrastructure, a focus of skills in the NT that industries can feed into and hand over their products for extraction and value add. That's going to involve investment. It's going to involve obviously an upskilling in terms of talent. But I'm going to show you how the collagen industry is going to change. If we're going to help the crocodile industry survive and many of its challenges, it needs to attach itself to a growth industry, such as collagen. Okay, so this is one way to do it at I guess lower cost to them, because they're not investing in their sole plant on their premises. It would be a cooperative in a sense. So this would allow us to produce a range of products which we would be essentially business to business. These are products that we would sell to cosmetics, to pharmaceuticals, to food industries. So that side of manufacture would take place elsewhere. And there's obviously precedent for that. Always look to New Zealand for innovation. And this is an example of the fisheries industry in New Zealand, which, has really maximized their use of waste around fisheries industry, not only fish carcasses, but also mussel as well. And they have a range of products that are available. Okay. All right. So how is the collagen industry changing? In about, I would say, 20 years, our collagen will probably come from precision fermentation and from genetically modified plants. And what's driving that is, I guess, sustainability in using large animals as a source of collagen. It's public demand for a more cleaner source of collagen. And so the collagen industry is faced with this transition, which will take some time, but you can see it in the ecosystem. There's a lot of startup behavior around moving to precision fermentation. And the collagen is one example. And you'll see that in Australia, particularly in Queensland and New South Wales, a lot more activity around using microbes that are genetically engineered to express protein at scale. And that's essentially what you would do with collagen, is use the human variant. It's very technically demanding, because you have to incorporate in those bacteria all of the metabolic systems that give you that functional collagen. So think again about the post-translational modifications. And in fact, plants seem to do that better. So plants seem to be a better route for high-end, post-translationally modified correct collagen for those high-end, high-value biomedical applications. So that would be my proposal, is that we move with the croc and the barramundi industry to establish traditional collagen extraction, but we're looking longer term to how we would look towards plant-based collagen in the NT. And this is an example of insulin. So insulin was harvested from animals. It's now made as a recombinant, and that is what we will see essentially with the collagen industry. We can see with many others. So this graphic just highlights how we would transition from the traditional skin-based industry, and incorporating that bioprocessing hub means that we generate a focus of knowledge, a focus of capability and infrastructure. And that then signals to the investors that this is where the next generation of the collagen industry would sit very well in terms of that ecosystem of skills. Okay, so we could call it a layered transition economy as we move. We're not leaving the skin industry. That will remain. There'll always be a demand, but we're ensuring the viability of that industry long term by linking to the traditional collagen industry and be prepared for changes in that industry as well. So I like this slide because it shows you how that we can still maintain, I guess, a really progressive and innovative crocodile industry, farming industry, by enhancing its circular economy at the base. So we have the basics of an income in terms of skin and meat still being viable. We then move towards the add-ons that I've talked about, oil and collagen, and we produce them through our bioprocessing hub. And then the future is about transitioning towards having the capability to, particularly about having facilities to grow plant-based collagen, but having our bioprocessing hub available to extract that plant-based collagen. Okay, so quickly then, so there are some challenges with fermentation in that it can at scale be very economically viable, but some of the modifications for collagen are not as precise in the fermentation as they are in the plant-based collagen. So to really have good modified post-translational collagen, we need that will hit the biomedical industry. We need to look towards a plant-based industry, and this table just highlights some of the differences between how we could use our products to enter markets at different economic values, essentially. So from the swim bladder and the traditional extraction from the crocodile, and then as we transition into the future with plant-derived to very high-end kind of biomedical applications in terms of implants and regenerative medicine, all of this technology, which will be apparent in terms of 3D bioprinting, which will involve collagen as a substrate, as we produce cellular environments for implantation into curing disease. Okay, so I'm sorry I'm rushing, but again, if we think of plant-based collagen, it's a much more, I guess, economically viable scheme for the NT because we can work on a modular system. We can increase scale by simply increasing in a modular system these growing environments. Whereas if we want to increase the fermentation, there's a lot of investment to do that. A lot of energy and capital is required for that. So there's a more of a stepwise approach with the modular. And so just to finish up, then there's something unique about what we have in the NT. There's nowhere else we can do that in Australia. If we can harvest these streams from the crocodile industry and the barramundi industry, we have a unique stream of collagen, and we can produce product that's essentially low volume. So we're not stuck with one of the, I guess, tyrannies of distance in terms of transportation of high volume products out of the state, and location as well in terms of Southeast Asian markets as well. Okay, so I'm just about to finish up. So this is just, I guess, my proposal. I kind of enjoyed that last bit. It made me think about where we might go with collagen, and it's very exciting. So we can improve the collagen industry in terms of its circular economy, add on with collagen and oil, and then build the capacity to make those products viable. But that will be the springboard, essentially the confidence in investors to come here to look to the next generation of collagen extraction in terms of plant-based. Okay, how am I doing? All right.
On time.
Oh, well. All right. Well, I think we'll just finish up with acknowledgements. So right now, the funding has come from AgriFutures Australia. So we're constantly meeting to see when the next level will come. And one is in currently with AEA Ignite program. So very keen to look to, obviously, cooperative environments where funding might come from industry as well. So I'd like to acknowledge Rena, who did incredible work in terms of all of the collagen work and really just promoted our work in other aspects as well, in terms of the communication. And then the team behind all of the work that we did included people from Curtin and also Michael Smat from JCU, who did a lot of the wound healing work. Sorry, I had to really rush through the wound healing, but besides the scratch assay, we did a very fancy assay that Michael does with around electrical impedance. So it's it made us see it in a lot more detail at low concentrations what those collagen peptides we're doing. Okay, so thank you very much for the opportunity, and thank you to everybody online. Questions? Please first. All right, you take it.
With the wound healing, you did with chemical control. How does it compare to, I guess, other species?
Yeah, so we looked at one of the easier collagens to get commercially, which is rat tail collagen. Yeah, so we did see a difference between the rat tail, but we would run a look at where the sources of collagen are coming from, so particularly bovine as well. Yeah. Yeah.
Thanks very much. Yeah, for the presentation, fascinating, and also for the invitation to chat about potential collaborations. Interested in the One Health teaching.
Oh, fantastic! Yeah.
But my question is about whether or not you've looked at potential differences in farmed crop collagen quality versus wild crop collagen quality, and considered the NTG crop management program, which has a consistent source of crocodiles that are from Darwin Harbour.
Yeah.
We use fatty acids and amino acids to look at food web changes in fish communities in the region. We see a lot of differences between habitats. So I'm just thinking.
Yeah, no, I think it's an excellent question.
Potential better quality collagen coming from these crop wild crops.
Yeah, because the picture I showed you.
That's the actual jumping crocodiles.
Well,
that's the wrong tail.
Yeah.
Collagen.
I mean, it's true. No, it's something we have thought about because, I mean, I'm sure there's a few in Adelaide River we've probably encouraged to be part of our research, but the farm crop is not evolving the same way in terms of mechanical load, but evolutionary that deposition of collagen would be the same. Anecdotally, I was interested in this, so I just wasn't aware as how feasible it was to use, I guess, trapped crocs as a source, and obviously they will be significantly bigger than some of the farm crocs as well.
That's an interesting one because the crocs that actually end up in Darwin Harbour are younger cohorts.
Oh, right.
They tend to get kicked out of the rivers.
Yeah.
And so they're actually probably not that dissimilar to the age.
Yeah.
They tend not to be the big ones, so it might be an interesting.
Yeah. But there's a lot of interesting questions. It's about whether that collagen has changed in some way or is more densely packed compared to the farm. But in other attributes in terms of oil or what's going on in terms of microbial antimicrobials potentially that might be discovered in the wild against the crop, because the farm crop will be on a very, sterile diet, etc. In that sense. My thinking was that there might be something evolutionary different in terms of the lining of the stomach, that we might find something in the farm crop as well.
You're planning to take some samples from crocodile.
That would be,
fatty acids and stabilized it. I don't know if there would be.
Yeah, we'll definitely have a check.
There's good collaborators at NTG that potentially help.
Oh, definitely worth having a chat.
Yeah.
Yeah. You had a question?
I have two questions. That's great talk, and I'm very interested to see all the discussion into commercialization process. So first, for one crocodile, how much collagen can you get, and how much is the market value? The second question is you introduce the superiority of the crocodile collagen. How can this superiority translate into the superiority health benefits of whatever?
So the second question, the function is all about the short peptides, and so there's a suggestion that there's a difference from the Tmax in terms of amino acid, and that will vary as well. So we need to look closely at the function of those peptides that we get from the crop in terms of compared to other species to look at that. The other advantage of the crop is the sources of collagen in different parts of the body that are not traditionally used. So in the bovine industry, it's primarily the hide. It's easy accessible. So to be able to access collagen from articulating joints or hyaline cartilage, that's a very specialized type of collagen, would have a very niche product and application in arthritis and things like very high value. The second question is harder for me to answer in terms of so if we were to calculate from what we got was about 10 to 15% from our starting material, which was about 15 grams. I'd have to relate that to the weight of a crop at term when they're harvested, which is about 2 years, I think, and then think about how much of that animal I'm taking for. So that's really at scale up, but it's the question that people would want to ask if this scale up is definitely viable. Yeah.
In terms of the extraction process, is the collagen similar to bovine or other?
Yeah. So the traditional method is the acetic acid treatment and pepsin digestion, and so that's a good point, because if you can develop a method that's different or improved, you can patent that, and that's quite important in the collagen industry. There's a company called Holista in WA. They have the patent for extraction of collagen from sheep, which they own. So if you want to extract collagen from sheep, you'll have to pay them a royalty. So part of the AE grant was to look at a different way of I guess concentrating the collagen through something called tangential flow filtration to try and I guess add something novel to the process that might be protected. Any questions online? Okay. Oh, good.
Cool.
All right. Well, thank you very much.